This is part 2 of the Chapter 12 lecture notes. What we were looking at here was what you would get if you crossed two true breeding purple flowered garden pea plants. So your pea generation would be both the parents would be true breeding for purple. And as you might expect, the F1 results would be all purple flowered offspring. And then if you crossed F1 siblings, which would give you your F2 generation, they're going to be purple as well. And that's really what you probably would have guessed if both of the parents were true breeding. But that doesn't teach us very much. Okay? So what does teach us something is a monohybrid cross between, for our pea generation, a true breeding purple flowered plant and a true breeding white flowered plant. So one of the parents is purple, one is white, and the one that's purple is pure for purple, and the one that's white is pure for white. They're not hiding any other versions of the flower colored gene. They only carry either the purple or the white gene for flower color. But what is interesting is that the offspring in the F1 generation are all purple. So, of course, one thing that we know is purple must be the dominant trait. That it must sort of like if an offspring gets um, a purple gene and a white gene, they're going to be purple. So we know purple must be the dominant trait. But the interesting thing, and where Gregor Mendel really began to be able to prove I'm sure Gregor Mendel probably had an idea before he started these experiments, um, but before he began to be able to mathematically prove um, the inheritance of traits, the, this next cross was the key. So when he crossed two F1s, the F2s gave him mostly purple flowered offspring, but some, in fact, it was around 1/4 every time he did this, and he did this many, many times. He got around 1/4 of the offspring in the F2 generation were white. So, looking at the notes, it says background in one experiment, Mendel crossed plants that were true breeding for violet or purple flower color with plants that are true breeding for white flower color. That's our pea generation. That's right here. And then that's basically what he's doing is called hybridization. He's creating a hybrid of the true breeding plants. And so the F1 generation were all violet. Every single one of the offspring in the F1 generation were all violet. But they were not true breeding. They were not pure. In the F2 generation, remember that Mendel looked at thousands of offspring at one time. He planted thousands of garden peas, and so he was looking at thousands. And every time he did this experiment, he continually got very, very close to a 3-to-1 ratio, 3 purple flowered offspring for every 1 white flowered offspring. Here's an example of the numbers. In one experiment, he got 705 violet flowers and 224 white flowers. And if you do the math, it's not perfect, but it's almost exactly 3-to-1. And if you think about why it's not perfect, is because this 3-to-1 ratio is a prediction. Um, it is predicted that you're going to get 1 white flower for every 3 violet flowers. And the reason that his numbers were so close to that prediction is because he planted so many garden peas because he got so many offspring. Now, if he would have only used humans or animals to do these crosses and gotten, like maybe 10 offspring at a time or one offspring at a time or just a very low number, the chances of him getting this 3-to-1 ratio in the F2 generation were not as great. But because he actually looked at thousands of offspring at one time, he was able to, the more offspring that you look at, the more likely that you're going to get that 3-to-1 ratio. But the bottom line is some of the offspring were white in the F2 generation. So what's going on? What are the conclusions that we can draw from these monohybrid crosses? Monohybrid cross is when um, we're doing a cross for one trait and the parents are hybrids. So the blending inheritance was not a viable explanation because none of the offspring were a blend between purple or violet and white. Inheritance appeared to be particulate. There appeared to be a particle that, like we call it now, we call it a gene, but there appeared to be a particle that carried the um, violet trait and a particle that carried the white trait. And the dominant version of each trait would mask the recessive version if you had an individual garden pea plant that was a hybrid that contained both versions of a trait. The dominant would mask the recessive. So the ones that are masked are called the recessive. So in that F1 generation, the dominant trait that we looked at was the violet color, and the recessive was the white color. And anytime that an individual inherited the violet trait and the white trait together, violet from one parent and white from another parent, they're going to show the violet color because violet masked the white color. Violet is dominant, white is recessive. So the recessive trait appeared to have passed through the F1 monohybrids unchanged to the F2. So the blending didn't really work because the recessive trait comes up again in the F2 generation. It kind of skips a generation. And we can think of examples in humans where a trait, maybe from someone's grandparents, skips a generation, um, and then the grandchildren have the trait. So maybe the children didn't have the trait, but it comes back in the grandchildren. We see that a lot, and that means the trait is probably recessive when it skips a generation. And the F2 ratio of 3-to-1, 3 dominant offspring to every 1 recessive offspring, suggested that the monohybrids must have carried 1 hereditary particle for purple or violet and 1 hereditary particle for white. What we would say now is that the F1 offspring had a purple gene and a white gene. Okay? So all of the F1s had a purple gene and a white gene. They were carriers for the white recessive trait. And ratios further suggested that the hereditary particles segregated into gametes and then came back together at fertilization. Segregated just means separated. And gametes, remember, are egg and sperm. So, question. Mendel had no notion of the physical basis of these particles. He had absolutely no idea what these particles were. So these particles turned out to be, I've said it, now I will write it, genes, and then the actual versions of the genes, when we talk about the violet gene and the white gene, those are actually referred to as alleles. So we would say the white allele or the violet allele. We wouldn't say, or like for seed color, we would say the green allele or the yellow allele. Alleles are just versions of genes. A gene carries the information for a particular trait. So the gene for flower color has a violet allele and a white allele. So that's how you would use that terminology. So now in modern times, this is the terminology that we use. Um, this terminology will get you through most of the questions. In fact, this page is probably the most important page. This slide is probably the most important slide for answering your questions and understanding how to solve genetics problems. So we use the term trait. A trait is a characteristic. That could be flower color, eye color, height, um, hair texture, curly or straight. I think in the last chapter when I did the thumb trait, whether it's straight or hitchhiker thumb. So traits are characteristics that individuals have. The phenotype is the trait that we see. It's the version of the trait that we see. So for garden peas, purple flowers, the actual color of the flowers is the phenotype. Now coming down, the genotype are the actual genes. So the parent, pure breeding purple, had two genes or two alleles for purple. But the F1 offspring had an allele for purple and another allele for white. So alleles, again, are the versions of the trait, specific versions of a trait or specific versions of a gene. Versions of a gene. So an allele can be dominant or an allele can be recessive. Now, when we do genetics problems, the dominant allele is going to be represented as far as our terminology, or not our terminology, but our um, abbreviations for different things. We're going to use for purple flower color, we're going to use a capital P because capital letters are dominant traits. And then a lowercase p for white flowers because lowercase letters are for recessive traits. Now, when we talk about the genotype, that's the individual genes. The phenotype is the version of the trait that the individual actually has, actually expresses. That's what we actually see. The genotype are the genes or the alleles that the individual carries. So if we say that an individual is homozygous or a homozygote, that means both of the alleles are the same. They are true breeding for that trait. So two big P's means that they carry both alleles for purple flower color. Two little p's means they carry both the alleles for white flower color. And then heterozygous individuals or heterozygotes are individuals that are hybrids. That means that they carry a dominant and a recessive allele, and we would represent that by capital P, lowercase p. One of the parents gave them a big P and one gave them a little p, and we know the phenotype would be purple, but they are actually hybrids for the trait of flower color. And then the zygote is the very first diploid cell that is produced when the egg is fertilized by the sperm. More terminology, the gene is actually the piece of DNA that codes for a trait. It is the specific piece of DNA in a chromosome that codes for a trait. So it's what Mendel referred to as hereditary particles. The locus is the place, the actual place on the chromosome where the gene is located. And alleles are versions of a gene. A reciprocal cross, this should be formal. A formal, a form mating cross where a previous cross is repeated, but the parents exhibiting versions of a phenotypic trait are reversed by sex. I think it's just formal. Okay, but anyway, it's just where you repeat a cross, but instead of the like instead of using the let's say